So normally, Tom Lair confirmed. Thanks, Chris. You can't get away with much in this crowd, in this audience here. So all right, we're going to make that the topic. So normally, Marcy introduces the speaker, but I thought since Marcy isn't here, I wanted to go ahead and sort of introduce myself. Not that I need to introduce myself, but I want to introduce sort of tools and technologies again. This is the first presentation for this year. We took a little break over the summer, so you could kind of break your pizza habit. But I also wanted to just kind of look at a few statistics. I kind of went back and looked and I went, how long has this been going on? We've been doing tools and technology. And I did not count pizzas, by the way. I should have, but that was going to be a little harder stat to get at. This was actually started way back in 2005, which doesn't seem that long ago, but if you actually look at the calendar, start flipping through the years, it's been going on for a while. And Danny Rhodes gave the first presentation January 5th, I think, of 2005. And a lot of this started because of NCIBI, and this was really the originator. It was a way that NCIBI did for presenting new technologies and trying to do a little more outreach. And NCIBI has sort of ended. Transmart has begun and some other things going on, but we kept the tools and technologies presentation because it really does give a chance for people to see some new things that are out there. It's a very diverse set of topics. There have been 220 presentations in all. They are all archived and available to watch. So Alex deserves a round of applause. Yeah. Because other than a couple times when Zach or Aaron or someone else has helped with, Alex has been here and sort of setting up and recording and has been here for all of these. So it's pretty amazing that we have that many available. That's the link. If you want to go back and look at the diverse topics, you can kind of search through by keyword and other things. And just so, you know, some personal egotistical statements, this is actually my 7th presentation on a unique topic. And it is the 221st presentation. Therefore, I have given 3.16742% of the tools and tech presentations. And Dr. Ullman isn't here, but he's always, and he will want to point this out, that whenever you give a presentation, to think about significant figures. Computers spit out very long stretches of decimal places which aren't really valid. So think about significant figures. Therefore, I've only given 3% of the talks.
Jim, you didn't figure your rank among all presenters?
I did.
Okay, never mind.
Thanks, Scott. Thank you for that segue. It's actually, there have been, of the 228 presentations, there have been 200 and, I didn't put it in the slides, I did it later, 247 total presenters out of 220 presentations. And of course, being in bioinformatics and thinking about math and numbers and statistics, I couldn't stop there, so I had to kind of go through and do this. There are 168 unique presenters, 247 total presenters, and here is the rank order. I only tailed it down to the beginning, but Terry Weymouth wins. Yeah, you can have that one too, Ella, either side. Terry Weymouth has given 9 tools and technologies presentations. Rich has given 7. I have now tied him for 2nd place. Alex at 5, Glenn at 5, Zach's given 5, Aaron 4, Manhong Dai has given 5. So we had quite a list of presenters, but the tail goes out quite a long ways. We have a lot of people that have given only 1 talk. And I think that shows both the breadth and span of the topics that we've covered. And I did a few statistics over there because Excel lets me do silly statistics. Okay. Today, though, the presentation itself is, oh, and by the way, for those that, I didn't do Marcy's standard opening. This is Tools and Tech. It is being recorded, and we have people live, a few signed in and listening from a distance. I'm Jim Cavalcoli, and there's probably not too many people in the room that don't know me. I'm assistant professor here. I'm also the director of the Bioinformatics Core, and I've been doing sequencing and DNA sequence analysis since before it was called Bioinformatics. That explains the gray and the thinning hair and everything else. So today I want to talk about kind of a neat topic. In the last year or so, there has been some technology changes and advancements that have really made sequencing small genomes kind of a game changer in the fact that how easy and efficient it is to do this. It's not that it was incredibly hard before, but there's a lot of cloning and sequencing and other things. And so I want to talk today about both the technology and some of the bioinformatics involved in sequencing, assembling, and closing small bacterial or viral genomes. Why is this important to do? Most bacteria and viruses in GenBank, if you look back 5 or 6 years ago, they were represented by single sequences being, this is what E. coli looks like, or this is what Homophilus looks like. And we didn't have a lot of speciation or variation or strain. There were some, and it was beginning in certain interest areas to do this. But bacteria evolve quite rapidly, and there's a lot of lateral gene transmission. So the word, although it implies different things in different terms, the promiscuity of gene transfer among bacteria is quite high. And so what you define as a species or a strain can easily have a gene or two or four from something else that it was with in an infection, something else it coexists with in nature. And so the ability to change these genes around and insert them into the genome is actually quite high. So the ability to rapidly and completely sequence a genome would improve discovery and maybe even epidemiology, tracking outbreaks, identifying drug resistance, and understanding the metabolism of a given bacteria. I'm not going to touch on today metagenomics or microbiome where you have a whole community of organisms. That still remains a little bit of a problem. But I really want to get at is sequencing an isolated organism. How many people here have heard of the Pacific Biosciences or the Pac Bio sequencer? Okay, so I won't go into a great deal of detail. I've got some pretty pictures here. But the Pac Bio is called SMRT, and that does not mean it's smart. It just says single molecule real-time sequencer. And it records and identifies fluorescently tagged bases added by a polymerase, which is bound to a well. And the DNA template circulates through this polymerase one base at a time. And it actually reads the incorporation of these by real-time laser excitation. You only get about 40,000 molecules per flow cell. And I say only because in comparison with things like the Illumina platform, which is sequencing hundreds of millions of reads, this is actually relatively small. However, and the single pass still has a significant error rate, somewhere around 11 or 12%. Yes, I didn't say 0.11 or 0.12. It is 11 or 12% on a single pass. However, because of the length of the molecules, no, I'll get to that in a second. Because of the length of the molecules has been increasing with the changes in chemistry, we can now sequence to very high fidelity molecules in the 2 to 3 kb range. And anything under that has extremely high fidelity. And so the length of the molecules sequenced by the Pac Bio machine is also, it's the only platform out there right now that's not in beta that will sequence these very long molecules. And I'll get to some of the statistics here in a moment. Right now, the average library insert size is running around 10 kilobases, so 10 kb, with an averaging sequencing length somewhere between 3 and 5, and 5 and 7, depending on certain library parameters that you can get for a single sequence from a single molecule. So how is this? The libraries are constructed this way. A given DNA sample, and by the way, it does not have to be a cloned sample. You can take DNA freshly isolated from an organism without having to subclone, without having to do any kind of amplification. If you can get enough DNA, you can sequence the DNA directly. The DNA is fragmented down to a certain library size, and this is a question of how you want to sequence this. If you go smaller, you get higher fidelity. If you go longer, you have the ability to scaffold and assemble long sequences that span repetitive regions. And that's one of the hard points to get past with Illumina or any of this short sequence reads, is anything that's repetitive sequence beyond the length of the read, you really can't assemble them uniquely. So they do an end repair, and then they actually ligate these dumbbell-shaped adapters onto each end of the molecule. And these each have primer locations on them. And what happens is the polymerase, the DNA is added to the wells, and I'm not sure the exact nature of this. I actually haven't been in the lab and do this. But the DNA becomes incorporated or bound to the polymerase. And the way it is, these waveguide, these are called zero-mode waveguide cells. They're actually a little metal cylinder that's about 50 nanometers across that has a window at the bottom that allows the light, a laser light, to excite the molecule and record the fluorescence. And then, because of the size and the exclusion of this, you either get 0 or 1 molecules per well. You can't have two. You can't have 2 polymerases, and you can't have 2 molecules in a well, and based almost exclusively on size. So this is a little bit more of another cartoon showing how this is bound. What happens is that as the polymerase processes in real time, so I think the recording is about, I think it's about 3 bases per second that are incorporated. I'm not sure the exact rate of this polymerase. But what's happening is the bases that are being added to the flow cell actually have the fluorescent tag on the terminal phosphate. And when the pyrophosphate is cleaved to incorporate them in here, the release of this then causes the ability to fluoresce, and that base is detected. And so as this is going in real time, flashing through, you can actually see this. And so if I'm going to attempt this to see if this will work. I don't know if everyone at home will get this, but I'm going to try. Oops. Wow, that's really. You know what? I think for sake of time, I'm not going to. I'll send these slides out. You can look at it. You can actually. This is a YouTube video that actually shows the whole incorporation process. It's about 5 minutes long, and I really don't want to go through the whole thing, but it is in here. What happens is the time in which it takes for this pulse to both occur and how long it stays around gives some measure of amount of time that it took to incorporate that base. And so the duration of the pulse is actually related to the type of base being added. And that becomes important for some base modification stuff I'll talk about in a minute. But it also allows you to get some idea of processivity and number of times a base is incorporated for homopolymer stretches as well. So the Pac Bio platform itself is ideal for a relatively small number of reads, around a length 8 to 10 kilobases at the moment. You do get some longer reads. The tail is quite long. We've seen reads out in the 30 kilobase range. The accuracy isn't incredibly high, but you can, if you're getting something that's 10 kb long and you can get 30 out of it, you can get a pretty accurate 10 kb read. And all of this information, including the qualities, is used in the assembly. You get about 40,000 molecules, as I've said. And really, the recent upgrades to the chemistry, which allows for a longer-lived polymerase in part, and the other features would be that you're getting a little cleaner incorporation of the nucleotides, and so that is going on. That chemistry change occurred in around April of this year, April-May. So what happens is, as in a cartoony sense here, the DNA fragments are capped. And what we get is the ability to read this transcript, because we know what the caps are, we know what the primers are, and so every time it reads through, so if you follow me and it's reading as it goes around the block, because the polymerase will actually go around those dumbbells and come back out the other side. So you sequence both strands, and the incorporation of the errors, even though it's a high error rate, is relatively random. And so you basically can auto-correct by sequencing over that same insert multiple times. And in this case, they call these reads of insert because they'll go through and you get the whole thing as a single molecule. Remember, one molecule in a well, so they know exactly where that molecule is. And now we get this read, that read, as many times around. So right now, you get 2 full reads of insert and a partial one. At this point, the polymerase dies. We get no more data out of it. And this happens in part because proteins have a certain half-life in here, and you're hitting with a UV laser about 20 times a second. So eventually, you're going to break down the protein. But here's a graph of some actual data. This is from an experiment that was done fairly recently, probably one of the most recent ones. This was actually an E. coli genome, and it was done to test the platform by a researcher here in the med school. And you can see out here around 30,000 base pairs. These are the longest reads. There was actually one around 35. But the majority of the reads are coming in somewhere in this 5 to 10 kb range, although it's quite a distribution. And you get some relatively shorter ones that sort of die off early. But the median distance between these adapters, when it measures it, this gives us an idea of how big the libraries are. We're going from somewhere between 5 and 10,000, with the majority of them right around 10,000. This is 10 to the 4th and is not readable on this graphic. And then this subread, or this read of insert length, shows that most of our reads of insert are actually peaking somewhere in this range. We've got a little bit of a bump here and some in this average, but most of our reads are falling in this 5 to 10 kb range. Again, so it's actually quite good data, and we've been actually increasing the size. And with reads that long, we can actually do an assembly. So that slide leads to this one, which is actually showing this is the coverage across. This actually isn't E. coli. This is a Klebsiella genome we were sequencing that was 5.2 kilobases long. And between the Pac Bio sequencing and another lab that was a collaborator had done some other Illumina sequencing, they merged the assembly and in a span of about a month closed the genome and submitted it to GenBank, wrote the note, and got it accepted. So they published this drug-resistant strain of Klebsiella that had been in ATCC. You could order it, but nobody had ever sequenced it. And so in the span of just over a month, they were able to do that. And this was right about the time that the technology was changing. And we ended up using 3 flow cells to get this data. So 3 flow cells, about 100, not quite 120,000, about 110,000 reads that it took to close that. And the coverage is actually pretty good. So we get pretty even coverage. Even though this looks kind of jagged, you get somewhere a little north of 50x coverage across the whole genome with those long reads. So we wanted to see what could be done with a single flow cell, because the company has been saying, and other people have been reporting, that a single flow cell, you can close a genome. And so we actually said, all right, let's see what we can do. And we ended up with 5 contigs for this E. coli genome. And it's a fairly standard E. coli. The sequence is out there already. So this was more of a test of what can you do. And we don't use any reference to guide this. This was de novo assembly by the Pac Bio assembler. There is a caveat here. The Pac Bio data comes out in an HD5 format that contains a movie and a lot of quality scores. And it's not amenable to a lot of other software tools. So you're not going to use BWA or Bowtie on the raw data. You can create these consensus sequences out of it to use in other assembly modes. But this is the assembled, the Celera. It's based on the Celera assembler called Blazor, and that's its basic mapper. Here was just under 5 megabases of genome assembled at about 40x coverage, 35 to 40x coverage here. And then there were these other contigs that showed up as well. And it turns out when, and I don't have all the blast scores here, it turns out that is the complete E. coli genome, and this particular one had 4 plasmids of various sizes. And so from this isolate, you were able to identify the different plasmids as well. And they're present at this one we're not sure is actually a plasmid. That's one thing he's still checking to see. But the others, we actually matched plasmids that were known to exist in E. coli. And so in that regard, we were able to get that assembly and get 5 million bases linearly assembled with no gaps from a single flow cell, which I think is kind of amazing. It's one of the reasons I chose to present this today, is that it's really, it's come a long way since even a couple years ago with being able to assemble this data. Likewise, this actually spans last spring, somewhere around the April timeframe, and we had a couple of flow cells in the old chemistry and a flow cell or 2 in the new chemistry to test things out. But this was a novel Providencia stuartii genome. It's a urinary tract infection organism. And this actually represents 3 flow cells kind of all glued together. You can actually merge the data fairly easily. In this case, we expected the genome to be about 4.2 to 4.3 megabases. That was predicted based on how close it was to everything else in the tree. And we got this contig 0, as it's called, which was just under 4 megabases. It was like 3.99. We're like, well, maybe it's smaller, maybe not. So we also got these others, and we thought, well, maybe plasmids, but let's investigate what comes out of this. And so the drawing on some actually older style of genome comparisons, it reminded me of my early days when we actually did dot plots to look at comparisons between organisms and from homologous regions. So if we compare on the x-axis out here is our experimental organism, and on the y-axis is this MRSN 2154. It's another strain of Providencia that was actually sequenced, and it was assumed that this would be very close to that reference. And in fact, it is. The start point is kind of immaterial because the circular genome could start at any point. This just happens to be where our genome starts here, and it's actually at the tail end of that organism. And then we've got here, but there's certain gaps where you've got DNA that's present in this organism that doesn't match anything in the reference or could be rearranged. A very large kind of insertion here. There's something missing or deletion in the experimental strain, and then these other insertion points up here. But it matched fairly well. So we wanted to see what these other contigs actually looked like. Contig 1 was actually fairly long, about almost 380 kilobases. And it turns out it matches this MRSN strain right around this 1.8 to 2 megabase or 2.2 megabase mark. And if we look around 1.8 to 2 megabase, there's this giant deletion, or what appears to be a deletion, in our assembled contig 0, the main strain. So sure enough, we're actually trying to figure out the orientation and the exact position, but it looks like this guy probably fits here. And I'll show you why it didn't assemble in a moment. That even though this platform's really good, there are still some pitfalls that are just natural biology differences. So what about the other contigs, just to kind of finish this off? It turned out that this one called contig 3 actually mapped back to contig 0, except for some gap in the middle. So something kept them apart. We don't know if it's a novel gene or not. This is still kind of under exploration. But it kind of mapped right back to it. So this we assume is contig 3 really belongs in the genome someplace and is fairly redundant. This little bit here is probably new. We're just not sure what it is yet. We know what. We know what the gene is, and it's actually not a Providencia gene. It's something that's come in from somebody else. We think it's Proteus or something along that line. And just to make sure that it wasn't like a plasmid or something, we actually tested against itself. So one thing that's good about dot plots is that you can actually blast a sequence against itself and see how well it aligns to see if there's any duplications. And in this case, in this particular guy, there was nothing. It was just that single line. It was itself. It's unique. Oh, and by the way, contigs 4 and 5, those other little guys out there, there's no match to known Providencia strains whatsoever, the actual genomes. It turns out that contig 4, if I do a dot plot and blast it against itself, you get these wonderful little corners that are indicative of the fact that that genome really is circular. And that's exactly what it would look like if you had a circular genome. And so when we do check it and see it is true, those last, in this case, it's about 6 or I don't know what it is, maybe 10 kb overlaps. And if you trim that and circularize it, you get a perfect plasmid, and it does map to one of the known plasmids. It's similar. It's not identical. There's been some changes in it. And likewise, contig 5 was a little bit smaller. This was about 50 kb, and about 10 kb on each end was actually overlapping. And again, so we have 2 circular plasmids. And they contain, in fact, this large one contains the gene they're very much interested in, which is a urease that they've been looking for that doesn't exist in the genome. So it's picked up at least one urease by this plasmid. OK, so I'm going to stop there. This is sort of the detective work that goes on afterwards. If you get a perfect genome, great, but there's going to be some detective work to identify what are the gene differences. And that's what makes it so powerful, is within a span of a couple weeks, you can get all of this data in which you can then go back and test, make PCR primers, verify things. There are a couple of tools, one I did not mention here, that allows you to actually take a genome like this that we've created and submit it, and it will actually map it against known, you tell it what you think it's close to, and it will go in and actually re-annotate all the genes and boundaries. So you don't have to do complete ORF prediction, but you can actually map it to the known ones and then go back and test the validity of those. It speeds up that process a lot. OK, so I'm going to jump to one other feature that makes the Pac Bio platform really, really interesting. And that is actually because if you're using unadulterated DNA, if it hasn't been amplified, if it's not been cloned and subcloned in systems, you can actually get a sense of the modification to bases that are present on your DNA, because the polymerase will pause differently and it will take longer or shorter to incorporate nucleotides when a base is modified. And I'll show you an example here that this comes from the Pac Bio site. If the adenosine is methylated, there's this long pause before the next signal comes out. A normal adenine or adenosine that's been incorporated will give a pulse that is pretty much in line with the other time sinks here. But a methylated adenosine, and this occurs for other bases as well, cytosine, methylation, and certain other base modifications that are shown down here. So the ones that they are actually publishing on their site is 5-methyl-C, hydroxymethyl. I'm not sure what FC is. I wouldn't expect it to be fluoro anything. So I'll have to look into a couple of these others I don't recognize. 6-methyl, 4-methylcytosine. There's an oxyguanosine and an oxyadenine as well. And all of these have different patterns of incorporation. And because the analysis software in here is actually looking at the movie in real time, it is actually can measure this time and see how often it occurs, and if it occurs in repetitive patterns, which I found to be really fascinating. Please note, if you amplify, like somebody wanted to look at methylation state in a gene, but they were going to amplify it up with a bunch of PCR primers, you can't. PCR does not incorporate modified bases normally. I mean, there may be ways to do it. I didn't research this deeply. But if you have normally a normal PCR mix, you won't get incorporation of the same 6-methyladenine there. You'll just get adenine. So you'll lose that signal if you're using any kind of modification. So the output from this, the first thing you have to do is you have to, if it's an unknown organism, you have to actually create the genome first, or you have to have a reference genome that you're going to align this to. And then you can take the reads from the DNA and align them to the reference using the software tools that are part of the Pac Bio suite here for modification and motif detection. And it takes about, for the E. coli we did, it took about an 8 or 10 hour run. It's not a short period of time because it's got to go back and map and analyze all the data, but it's not too bad. And then it will give you these great colorful outputs that I'm still learning to interpret. But basically what they're showing you is for certain patterns, in this case GATC, in which the A, and we've lost the print font here, but in which the A has been methylated, you get this interesting pattern of outlier of the behavior that's really standing out. And then there's another one, C followed by five A's? 4 A's. And then another C. That's another one that has a pattern that's distinct from the others that are here. And it then summarizes these graphs into a nice table, which I found to be much more readable. In this case, GATC, in which the A is a 6-methyl A, actually it uncovered 60,000 motifs that it detected this pattern in out of 60,600 that it found in the genome. So 99.8% of that, every time that GATC is there, it's got a 6-methyl A. And in this case, it's 99.8% also for the CAAA, with the last A before the C being methylated. And both of these patterns, the first one explicitly, this one is less well known, and I haven't been able to find any literature on it. But in this case, the GATC is part of a restriction class in bacteria where they methylate their own genome so that any kind of invading phage or other organism can be subjected to restriction digest of invading DNA, and it won't degrade its own DNA because the methylation is resistant. And so they methylate these patterns, and then they develop a cleavage system to cleave that pattern when it's not methylated. And so it's basically an immune response, if you will, in bacteria, and it's called a restriction class base modification. The reason we did this was actually we did the Klebsiella genome I showed at the very beginning. They completed this, they published it, and within, I don't know, a week of the publication going out, got a call from Rich Roberts, who is, I don't know if he's either senior scientist or CEO from New England Biolabs, and I didn't realize at the time, Nobel laureate. I don't keep up with all of them. Not that many, but I really just don't keep up with them. Who've said, we'd like to see the methylation data because this is really important. And Mike Bachman said, we didn't run it, and thought that was the end of it. He's like, no, you have the data. You just haven't. All you have to do is run this analysis. And then he came back to me and said, like, how much is it going to cost? I said, I don't know, maybe an hour of my time. We set up to do it, and the data came out within 8 hours of starting the run. They've since sent it back to him. We haven't heard back yet. We just sent it in last week. But they're looking for new restriction class patterns here, or things for new enzymes and things to discover, and et cetera. So his point was, we want to start making this mandatory when you submit bacterial genomes that have Pac Bio sequence data, that you start submitting these methylation patterns, because the data's there. You've discovered it. It's just that you're not publishing it. So we may start doing this as more of a standard thing to start to look at these classes. Right.
Is it possible that the motif is difficult for the polymerase to put together, and that introduces the delay?
I don't know. It could be something about the secondary structure there, but since it's adding one base at a time, you're getting that delay at every point in that base. And this is something that if you were if you'd studied restriction, if I had studied these restriction class endonucleases, this is a known pattern. It's a very common one. So it's possible that you're discovering what's really there. I don't know if it's an artifact. We'd almost have to take an organism that has that pattern and not that class of restriction and see if the methylation was there. Pac Bio's done a fair bit of work on this. I just haven't. This is fairly new to me.
I guess it would always be the same, probably. And then
yeah.
Then you'd see it.
Yeah. It's not really. It's too short to be any kind of hairpin structure or anything else in the DNA itself. It's too short. 4 nucleotides is not long enough. And there's nothing about the pattern on any side. These other ones down here that it finds at a very much lower level. At some point, there's a spurious noise where you're finding motifs and you've got some of this, and it's not high enough. So their cutoff is usually around this modification, this QV value, and they don't explain that well. Under 30, they sort of say, yeah, noise. And the longer you go in the motif, the more likely you are finding a spurious event as well. So as those kind of converge around the lower quality and the longer motifs, they're probably less likely to be real. That's what Pac Bio claims anyways. So. Okay.
So quick question, Jim.
Yeah.
I guess more about the nitty gritty.
Sure.
So as you said, because most of this is read in from a movie, it's mostly using Pac Bio software to analyze this, right? So when they go from the movie to sort of these tables and figures, is it straight to these tables and figures, or can you actually get sort of like maybe a semi-raw readout of the time between nucleotides? And I mean, do they go straight to say, oh, this is methylated, or do they actually provide you the timing between the nucleotides, and then you could derive your own statistics from that?
The current software, you don't get those intermediate files in their web portal.
Yeah.
They have sort of raw data, and they've got a series of intermediates, but they're not really intermediates. They're mostly these graphics that have already been derived. But there's a whole underlying tier of command line programs, a lot of Python and scripts that they actually combine together in these pipelines. And so you can go into the individual ones and get that data out.
Okay.
It's just that it's not easy to do. You'd have to really almost have to read the code. Their documentation on those subprograms is almost non-existent.
Okay, but that is not. But that is something that can be done. I guess.
Yeah, and
one of the things, like go back to like complete genomics, and one of the issues that that there was that it was sort of really hard to analyze the raw data. You had to deal with their sort of their variant calls and their statistics, and there wasn't a lot of bioinformatics software that it was developed around this, as you have with Illumina. And,
yeah,
and we see where that goes. When you don't have the bioinformatics support, it's sort of people like to tinker with stuff. Yes,
and the Pac Bio software is more black box than I would like, partly because it's the lack of good documentation. This is a company that did a really good job of developing the technical platform, and the software. And this is an adage that I've stood on this for the last 15 years, that companies that build hardware should not write their own software, and software companies should not build hardware. I mean, there's just the twain shall never meet, and they don't do it well. It's a frustrating point because they're really good. The platform itself to get this data is really, really nice. There's some little glitchy stuff in it, but it works really well. Their software, on the other hand, we've run a lot of wrestling matches with trying to get it to work right. It breaks at odd places sometimes, or they'll say, oh yeah, but you can do that at the command line, and the program file names are all different. You really have to sort of like read their own code.
So these are bioinformaticians who wrote the code, not software engineers. So this makes sense.
Point taken.
I'm calling the pot kettle black here. Yeah, it's like looking at my own code. But.
Yeah, it is like that. I actually have a friend of mine who's a software developer engineer that writes code for a living, and he when he took a look at the website I showed him for this, and I'm sorry, Pac Bio, putting this on record. He said he referred to it as dog food code, because after as an engineer, as you've written code and it just gives you a debug report, and you get used to it, but pretty soon you get used to eating your own dog food and you don't notice anything wrong with the software. And so a lot of this stuff was released because there's just lack of good user interface on a few points and things like that. But they're making improvements, and the underlying stuff is there. The other thing, which I am patiently waiting for, is that this summer at ISMB, Gene Myers gave a presentation, a keynote, and in it he talked a lot about Pac Bio, and he mentioned a lot of software that was going to come out of his shop this fall. And it's fall, and they're not done yet. But they actually specifically wrote things for putting different software applications out there for analyzing this type of data. So I'm hopeful that now with some competition, it would be like, no longer do you have to use ABI sequencer. There's all these other things that you can now use. But I think the data is really cool for this kind of niche work. Jeff.
So you mentioned a web portal. Does that mean you're submitting data for some of these analyses to their place?
No, they created, their portal is a combination of their Python code, a web interface, and a MySQL database. And so it knows where the raw data files are. And you have to go into this portal and you have to load your data. You have to pick your applications.
Locally run.
It's run locally. Yeah. We're actually in the process, right now it's run out of the sequencing core on their storage and our server in the Bioinformatics core. Pretty soon we're going to move that apart and set up a virtual machine that will actually use Flux in the background because the number of processors, I can't throw enough processors at this, and more people are using it. But it is run locally. We don't have to send data out. We do have a problem with the way it's set up. Again, hardware people that wrote software. Their level of group control and access is sort of like nobody, one person, everybody. There's not a lot of levels of group control. And so there's stuff in there where, since I have view of everybody, and that's how they set it up, I can see projects that we're not even working on. And I can download that data. And there's no restrictions on it whatsoever. So we're trying to get to a better level of granularity with this new virtual machine and some other stuff we're doing. But right now we're running on the honor system that we're trying to create usernames for a lab and let the lab group see only their stuff. But some of us still have to see everything in order to manage the system. It's not.
Anybody doing clinical samples?
No. There's actually been, to my knowledge, no human samples other than a single gene amplicon that were run. And it was just a single gene, probably not HIPAA identifiable at that point. And we do know who can see it. The problem is a large number of people can see it. And that's not what would be good for any kind of HIPAA or clinical things. But we do know how many, and we know who they are. It's just too many. Okay. So the other interesting thing is that actually running a sample through is about $450, $460 to actually just get it through and get the data. And we put in about 2 to 3 hours to get the assembly done and basic annotation. So it's really not that expensive to run a sample through. If you have a new isolate or strain, you're probably talking between $700, $800 per sample to get full sequence assembly for a strain of interest. So I just throw that out there so people have some idea of what the dollars are like for that. And I think I'll stop there and take other questions and stop. Thanks.
So you just mentioned that you can use you can detect the modification of nucleotides based on its layered signals. Or you also mentioned many kinds of modification. Is there any distinct difference between all these kinds of modification?
There must be, although they don't go into great detail on their website. I think what they've done is they've looked at training sets where they've done that kind of modification to DNA and then run it through and looked at the signal processing part of it. And there must be a way to distinguish between certain signals. So perhaps if a delay is of a certain type and a duration, that it consistently is a 5-methyl cytosine, whereas a 4-methyl cytosine has either a shorter or longer. And there must be something to separate. They don't go into great detail on their website as to how. I did have a request from a researcher here on campus. They went ahead and modified some nucleosides for RNA incorporation. And they wanted to run Pac Bio. And Pac Bio was interested in doing this, but it's been like 8 months and they haven't heard back. They said, can anybody else help us? And I went, no. Because right now there's nobody that I know of here that could actually tease apart that signal processing. I pointed him to Fan Meng and I pointed him to a couple other people that could possibly do this. But right now we're having to rely again on the black box of whatever Pac Bio has said that this is a 5-methyl cytosine or a 6-methyl adenine or adenosine.
Another question is, in order to get a reliable sequence of certain samples, how many full processes you have to run for each flow cells?
Can you rephrase that or?
What I mean is like at the very beginning when you explained the sequence process, you said like it's iteratively going over the circle again and again, right? To fix the error rates. To fix the errors. So how many full passes you have to run for this? Full cycles you have to run this?
We put our filter at 3 full passes primarily. By default, it's 1 full pass, and I always change that. But 3 full passes gets you probably about 95.5% accuracy. And otherwise, you're beginning to, depending on how big your fragments are, that is plenty accurate if you have 30x coverage across a genome. Because if you're stacking up 30 high and you've got 95% accuracy, you will have some base mismatches, but you also get much larger read length. If you want high accuracy, we've done some very high filtering where we've gone as high as, the website will, again, a hard-coded feature that doesn't need to be there. The website allows you to only go to a maximum of 10 passes. I don't know why, it's kind of arbitrary. But if you go down into the command line software, you can change that number. So we've gone as high as 25 passes where that was the median and said we only want data that's gone around at least 25 times. And the accuracy didn't improve after a certain point. What's that?
Was it 10?
It depends on the sequence, and maybe there is a reason why there's a 10, because it really doesn't matter after that. No, by mathematics and statistics, the error rate should continue to improve, at least asymptotically. But what we found is that the amount of PCR errors that went into making the amplicons built up far before that. We were trying to get down to 0.1% error. Pac Bio claims that at the highest rate, you get down to about 0.5% error, and between 0.1 and 0.5%. And we couldn't get anywhere below about 0.5. And in fact, I'd hesitate to go less than 1% error because of PCR incorporation. I have some unpublished data from Sami Malik, actually Brian Parkin, where we looked at, they made amplicons across a tumor gene they were interested in. And actually the most error came in the primers they bought. We were seeing at certain bases in the primers, we were seeing 2.5 to 4% error of incorporation of base, the same base, over and over again. So you were seeing a C at this position, and it's the third position of the sequence. And we went, wait a minute, that's not the gene, that's the primer. And so after PCR incorporation, either it's the PCR misincorporating always at that base, or the primers you bought are dirty. So, I mean, you could use this to really evaluate very low levels of error and things as well, because of short sequences like that, you can easily get 10 on that short sequence, we were seeing a median number of passes at 25, 26, something like that. That's why I set it as high. I want to say, okay, what's the top half look like? And it didn't get any better. So.
Jim, how big are the files? So you mentioned that we're going to implement this on Flux, which the battle with Flux is getting the data, raw data over there and getting it back. Is that a problem?
No, these are fairly small. They're in the megabyte range because you're only dealing with 40,000 sequences. So you're not dealing with anywhere near the file size. They're 5 to 20 megs, depending on how many flow cells you're mixing together and things like that. So no, and moving 10 megs, not an issue. Yeah.
Have you done RNA yet?
We have not. I think people have run it. I just don't know. We've not had anybody actually run straight up RNA on it to see, because by default, it's a DNA polymerase that's in there. And I just don't know if anybody's run, if you have to change if they've got a flow cell with different polymerases on there to actually handle the RNA. Yeah. I think people have done it because I've seen some things out there, but I have not really studied it. It may be an interesting way to look at it. So. Okay. All right. Thank you.
Jim, 5 formal, say